So thank you for asking me to come. I mean, we've been funded by the MRC for a long time now and very grateful for it. Indeed, everything I'm going to tell you today wouldn't have happened without the MRC, I think. So my theme is birth asphyxia. This is a terrible condition. It's been well said that the most dangerous time of your life is the day you're born. A wit pointed out that it's not quite as dangerous as the time you die, but actually, in statistical terms, it is one of the most dangerous periods of your existence. And every year, about 5 million babies die before their first birthday. 2/3 of those will die in the first month, 2/3 of those will die in the first week, and 2/3 of those will die in the first day. So the day of birth is a very dangerous time. And actually, it causes more deaths than people realize compared to other childhood diseases. So without making too much of this point, birth asphyxia accounts for something like 9% of the world's global deaths in children, and that's more than malaria. And people don't often realize that. And of course, the majority of these deaths occur in the sub-Saharan part of the world, the southern hemisphere, sub-Saharan Africa, South America, and the lower income countries. And in those countries, a large amount of the attributable ill health in children is also associated with birth problems. Cerebral palsy in Uganda has been looked at by Jane Hassel, a very good study. And she reckons that about 60% of the children with cerebral palsy have an intrapartum related encephalopathy, or as we would call it, birth asphyxia, with or without infection. Now, in the UK and most developed countries, the number is probably half or slightly less than that. So birth asphyxia probably accounts for between 20 and 30% of the cerebral palsy in the UK. But that's still an awful lot of children every year who suffer lifelong handicap because of their difficult birth. What actually goes on when you're starved of oxygen around the time of birth? Well, the placenta doesn't transfer oxygen properly, and the body has many coping mechanisms with that, but they eventually give up. And the end result of it is that you suffer a global hypoxic ischemic damage to the body, and the brain develops significant problems. These are two MR scans, one of a normal baby around the time of birth, and you can see on the left-hand slide a nice normal scan with an arrow pointing to a very typical normal feature, which is the posterior limb of the internal capsule, which shows up very brightly. And on the right-hand side, a child with severe injury, and it doesn't take a neuroradiologist to see that there are big white lumps in the middle of the brain. These are actually infarctions in the basal ganglia, the thalamus and basal ganglia. And these are probably best thought of, especially the thalamus, as being the internet portal of the brain. This is where all the signals go in and out of the brain. Everything you feel from your body goes in through the thalamus to the cortex, and everything you think is transferred to action through the thalamus. So if you knock this out, you actually knock out the communication center of the brain, and the results, not surprisingly, are absolutely devastating for outcome. And children who suffer this injury have cerebral palsy of the most severe kind. This is not just a mild insult. This is terrible, life-impairing handicap. Life in a wheelchair, not being able to feed yourself, communication very limited. These are very severely affected children. So what's actually going on when this insult happens? Well, at this point, I'm afraid we have to do a little bit of biochemistry. I promise you this is all the biochemistry we're going to do. And it's at the level at which I understand it. So I think it'll be the level that everyone can understand it. Basically, all the processes that your cells undertake, everything your body does requires energy. And that energy is used in the form of adenosine triphosphate, or ATP. And you normally make this by, in a very effective way, by breaking down glucose, and you have mitochondria which turn that into energy. And that works very effectively. But there is a problem. You only have a few seconds worth of ATP in any cell of your body. Now, from an evolutionary point of view, this is actually a bit unstable. If you're a caveman running across the savannah being chased by a saber-toothed tiger, and you don't have a Mars bar in your pocket, then actually you run out of glucose very quickly, and you would run out of ATP. Fortunately, nature's not stupid. It does have a way of dealing with the fact that when that system doesn't happen, there's a backup store. And that backup store is actually phosphocreatine. Normally, ATP is broken down to ADP, and it gives off an inorganic phosphate atom. But if you can't make ATP the normal way, the body can actually make ATP a different way by turning ADP into ATP by breaking down phosphocreatine and giving off creatine. That backup system is essential, and actually, by a quirk of nature, it's something we can observe in the live body using magnetic resonance spectroscopy. And on the right-hand side of the slide, you can see a magnetic resonance spectra from a human brain, actually a baby's brain, showing several peaks. These peaks all relate to concentrations of chemicals in the brain. There are 3 peaks for ATP, 1 for each phosphate on the ATP, and 1 for phosphocreatine. You can see there's lots of phosphocreatine in this brain and not much in organic phosphate. This observation was made by Osmond Reynolds, who you see in the picture here, who was an FRS and the professor of neonatal medicine at University College London, and the most important person in the story I'm telling, because he made this essential observation shown in the slide. He arranged for people like me to put a large number of very sick babies into an MR scanner. This was novel at the time. The first MR scanner was put into UCL to do this in the 1980s. And we took down a lot of babies who were all very sick and put them into the scanner. This was clinically a difficult undertaking, but actually it produced a very interesting result. The graph you see from an old DASO slide that Osmond Reynolds made is showing days after birth on the x-axis and the amount of phosphocreatine expressed as a ratio to inorganic phosphate, which actually is just one way of doing it, on the y-axis. So amount of phosphocreatine on the y-axis and days after birth on the x-axis. The 2 horizontal lines are the mean and lower confidence limit for the normal values for inorganic phosphocreatine. And you can see that it falls down. That's a very clear evidence that the body can't make energy the normal way and that the normal energy processes are damaged. Actually, it's mitochondrial damage that causes this. But the really important part about this slide is not the fall, which is important enough. It's the fact that the first data point at about 1 day of age is normal. It's in the normal range. And this was utterly surprising to everybody who saw it. And indeed, not everybody believed it. Again, I would say these are all children who've been born after significant birth asphyxia. They've been untimely ripped from their mother's wombs, many of them, by a well-meaning obstetrician, and they're on intensive care in the neonatal intensive care unit before they're scanned. They're often fitting, they're on ventilators. These are not normal children, and yet, their brain energy metabolism, their ability to make energy is normal for several hours before it declines. This observation made it very clear that there is a lucid period during which it is sensible and rational to think of intervening to try and prevent the brain cells from dying. They basically don't die for a few hours. Now, this is quite a long story, and actually it turns out that these cells are all dying by apoptosis, but I won't go into that. The point is that we have an opportunity to try and do experiments and try out treatments which would prevent this later decline and this cell death. You can see this in another way. This actually is now an MR scan, not of a baby, but of a piglet, and this is the top of a piglet brain. It's one piglet in a scanner where an artificial ischemic insult's been made after the baseline. And this is diffusion imaging. And in this particular type of scanning, where the brain is light colored, it's normal, and the cells are living properly and working properly. And by the time it gets black, these cells are dead. And again, you can see that in the baseline scan, the brain is light colored, all the gyri are nice and clear to see. But by 10 hours, there's a little black worm crawling across the cortex, which by 14 or 17 hours has become a snake. And by 45 hours, the whole brain is black. This brain is dead. But at 3 hours, the brain is normal. We have a chance to intervene and do something during that period. Well, we tried all sorts of different things, none of which worked until we tried cooling the brain down. And to our surprise, cooling the brain down after the ischemic insult worked. And this is a rather complicated slide and probably the most complicated one I'm going to show. But there are 3 groups of piglets in this slide. On the x-axis is the time from resuscitation, which means that the piglets were all suffered or mostly suffered an ischemic insult. On the y-axis is again phosphocreatine, which I've just labeled as energy metabolism in this case. And the 3 groups are piglets who had the insult that you saw in the previous slide, where the brain slowly went black over the course of 45 hours. One group of those piglets was kept to the normal temperature. Another group had their brain temperature reduced by 2 or 3 degrees for 12 hours following the insult, and that is shown on the graph as labeled as cooling. There's a control group to whom nothing happened. What you can see in this slide is that the insult causes the energy metabolism of the insulted piglets to deteriorate significantly. Then it returns to normal, and we have our lucid period again. this is effectively the same as the baby being delivered by the obstetricians and taken to intensive care. In the normothermic group, the energy metabolism then declines and these cells start to die. But piglets who are cooled down for 12 hours do not see this decline. They don't have this deterioration in their ability to make energy, and they don't have this level of cell death. And just to make sure that things weren't being delayed, as opposed to prevented, the hypothermic group was carried on for an extra 12 hours. So this was the first experiment of many, which showed that cooling the brain down after an insult could protect it from brain damage and stop this secondary energy failure, the secondary decline, which causes long-term damage. Well, of course, the attraction of cooling is that no drug company owns it. There's no patent on it, and you can do it without having to pay anybody for or get access to a drug or any other type of equipment. It's very, very simple to do. So it's very easy to run a clinical trial. So this is one of our first clinical trials. This is a little baby called Hollie Marks. Poor little thing was born after a very difficult delivery, taken to intensive care unit, and then cooled down for actually 3 days for various reasons. We went rather longer in the babies than we did in the piglets. And this baby, as you can see, is on a ventilator. You can see the ventilator tube going into the mouth. You can see all the other accoutrements of intensive care there. I won't bore you with what they are, but the big thing over her is a big blanket, which is a paper blanket with holes in, blowing cold air. And we reduced Hollie's temperature by 2 or 3 degrees and just waited to see what happened. Well, she wasn't the only baby that was studied. We did 2 trials. The Americans did another trial. And the end result of those 3 trials is that actually, if you cool babies' brains down for 3 days, you can significantly reduce their brain damage. And I'm going to show you that data now in what's called a forest plot. For those of you who aren't familiar with this, the various numbers of trials that looked at hypothermia and tried to see whether it would work or not are listed. CoolCap, NICHD, Toby, Zhu, ICE. CoolCap and Toby were our trials. Toby was the MRC funded trial, and you'll see it's the biggest trial there, and actually it's the definitive trial. Then the blue dots show you what the result was, and the result was either better with treatment, which is the favors treatment side, or better with control, which is the other side of that vertical line. And all of these trials, as you can see, were better when the babies were treated. So this reduced death and disability at 18 months of age in these children in all of the trials. Not all of them were significant, not all of them were big enough to be significant, but actually overall, this treatment was highly effective. One of the big worries that we had setting this all up was that we would keep children alive who would then go on to have this very severe disability that I described earlier and very difficult lives. Fortunately, this treatment doesn't just stop children from dying, it actually improves the outcomes for children who survive. And you can see here that in the 3 main studies, CoolCap, the American study, and Toby, where the numbers are big enough and the outcomes are big enough to follow this, we can see that there's a reduction in cerebral palsy, reduction in severe disability, reduction in blindness, and in other measures of outcome. So it's keeping children alive, and it's making the children who survive have less disability as they grow up. So once these trials were done, it became important for the UK to start thinking about doing this. The Americans started immediately, you'll understand. The Americans are fast adapters, and within several weeks of the first trial, there were adverts going out on radio saying, has your child got cerebral palsy? Were they called at birth? If not, call such and such lawyer's office. But the UK doesn't do that. The UK is very careful. The UK didn't start developing cooling protocols for the whole country until the trials were complete. And when they did that, the British Association of Perinatal Medicine and other people got together, wrote the protocols, and you can see here that over the course of a couple of years from 2006, just after the data came out to 2010, there's a slow increment in the number of children who are cooled. And by the time we get to 2010, we're cooling about as many babies as we think need it in the UK. So that's the NHS at work. And I think it's a good example of a planned, effective introduction of a therapy. This is the register that we set up to follow it, which accumulated the data to show this. Does it have any effect on the real outcomes? Well, this is a graph which I took from the Institute of Health Data Metrics in Seattle, showing the number of disability lost life years, so disability adjusted life years lost in the United Kingdom caused by birth asphyxia. They call it neonatal encephalopathy for various reasons. And you'll see from this graph, which starts about 1990 and finishes about 2017, that there's a significant fall going on during the 1990s and early 2000s. And that's, I think, because of better obstetrics, better access to care, more money going to the health service, better nutrition, just general improved standard of living, all the things that reduce perinatal mortality around the world. But when you get to 2002, sorry, there's a bit of a horizontal period here. And cooling was introduced at this point here, about 2006. And after the slight horizontal period, you can see the number of disability life years starts to fall again. And this is the period when cooling was being introduced in the country. Now it's leveled off again now, I think that's fair to say. But over that period, there was a significant fall in the number of disability adjusted life years lost in neonatal encephalopathy. I wouldn't want to claim this is all due to cooling, but I would like to think that cooling plays some part in the improved outcomes at a national level. It's not just the UK that saw this. The same sort of phenomenon happened in the US, which you can see on the left. And I chose Finland to look at because Finland has famously good perinatal care. And it's not so clear in Finland. They have lower levels of disability anyway caused by neonatal encephalopathy. But there's a suggestion that maybe it happened there as well. And this is Hollie Marks going to school. She grew up, this is her in her school uniform on her first day at school. But actually this begs a question, which is, does this effect, which we've shown to be effective at 2 years, carry on? Can we show that it carries on? There are well-known examples of treatments where you treat a baby, the effect is seen at 2 years, and by the time they're 5 or 6 years old, there's no difference between the control group and the treatment group. So we followed our trial up till the children were 7 or 8 years old. So did the Americans, which was great. So we had 2 studies that we could look at the outcomes for. And this again is a forest plot, again showing these 2 trials, this time showing the outcome at 6 to 7 years for the Toby MRC funded study and the American study. And in both of these cases, there's an apparent improvement. The NICHD study was too small to show a significant improvement. Toby did show a significant improvement. And together, the total group, there is very clear evidence that the treatment favors hypothermia. Hypothermia has better outcomes than controls. The numbers needed to treat for this treatment is about 7 or 8 to reduce 1 child with death or cerebral palsy. That means we have 7 children coming to the unit, only 1 of them will actually get better. That sounds like a really bad ratio and really bad outcome. But in fact, it's not as bad as it sounds, because if you think about statins, which are the drugs that reduce heart disease and stroke, I mean, are probably one of the biggest advances in medicine in the 21st century, 20th or 21st century, until the vaccine for COVID. The numbers needed to treat for statins is 100. To save 1 heart attack, you have to treat 100 patients. So treating 8 patients to get 1 outcome is actually, in medical terms, a very good, very strong effect. It also improves quality of life. This is the quality of life at 8 years for 6 or 7 years, sorry, for the Toby study, and the hypothermia group have better quality of life. They also have less disability, and in fact, importantly, they have less severe disability. This is what really matters, I think, in this treatment. And the societal benefit is also financial. If you do a very quick estimate of the cost of looking after a child with cerebral palsy in their life, it's about $1 million, and that figures come from the US and from Denmark, so it's not just a US figure. If you then estimate the number of children that have been saved from needing lifelong care by cooling, you work out that we've saved the country nearly $1 billion, nearly £1 billion over 10 years. That's a very rough figure, and I wouldn't want to stand up in court and defend that. But actually, we did do a very careful healthcare cost analysis, which showed that in the Toby study, it's about £1,000 cheaper to look after a child with hypothermia than not to. And also that more children in the hypothermia group have parents who can go to work. And that, of course, is a major benefit to the economy. The major benefit, actually, from hypothermia for the country, it comes from reducing long-term disability and the need to look after children. Does it work in low-resource environments? Well, one of my PhD students, Nikki Robertson, who's now a professor in Edinburgh, went and did a trial in Uganda, which didn't show any great benefit. That's probably because there's 2 different things about Africa. One is there's many more infections involved in the encephalopathy, and also the labors are very, very long indeed. We think a long labor is 12 hours. They think it's 12 days. So that, plus the fact they couldn't do neonatal intensive care along with the cooling, meant that it didn't work very well. But there are now trials going on in a more rigorous fashion, and we hope that we'll show that it works in low resource environments as well. Because of course, as I said earlier, there's no drug company with a patent on this. It can be made free to everybody. It's certainly being used in middle income countries. The National Children's Hospital Hanoi has just published a very nice paper showing a very simple way of cooling using a phase changing material instead of a complex cooling device. And in South Africa, they're reporting cases in some of their more difficult to reach hospitals. So it does work in middle income countries. It's also a proof of concept of something which is very important. It's not just that cooling is a treatment that we can give, it's that it shows that treatment is possible. It's a proof of concept. It's not perfect. It doesn't save everybody. There are still children who have terrible outcomes despite being cooled. But it does mean that we now know enough about the biology of birth asphyxia to know that we can rationally try different drugs and different treatments during that lucid period before the brain cells start to die, before second generatory failure, and try things that will add and make cooling better. So these are a list of drugs that are being repurposed for asphyxia. The MRC funded us to do a trial of xenon. Xenon is an extremely effective way of preventing brain damage in rats. It's also very good in piglets. It's very unfortunate it doesn't work in humans. And that's a very sad outcome for a 10-year research project. But it's not the only one. Erythropoietin is currently being looked at by our friends in Seattle. They're doing a fantastic study there, and we'll have the results out in a couple of years' time. And there are a lot of other drugs which are all being repurposed. My particular favorite at the moment is Extendin, which is a diabetic drug. And actually, there's real benefits in looking at diabetic drugs in this context, which I won't go into. But I think it's worth just finishing by saying this is Hollie Marks going to university. Hollie is studying physics at Edinburgh. When I was at school, you weren't allowed even to do physics unless you were very, very clever. So I think that she's actually, I wasn't allowed to do it. I think she's done very well from a little baby who was being called on a ventilator 21 years ago to doing physics at Edinburgh. And I just want to thank the MRC for funding most of this work. They funded the Toby trial, they funded the Toby children's study, they funded the ill-fated Toby Xenon study. And while the Wellcome Trust has actually funded quite a lot of the more basic research, we couldn't have done what we do without the MRC. My centre was set up by the MRC about 10 years ago, and we've been hugely grateful for the support we've had. And I also have to recognise my colleagues, because I didn't do this work by myself. My colleague Dennis Asapardi in the Centre for the Developing Brain actually ran the trials very effectively. Osmo Reynolds, I mentioned already, is the progenitor of all this work. And then friends and colleagues across the world, Peter Gluckman, Alistair Glunn, Mariana Torres and Eddie Whitelaw, and many, many others who actually made this all work. And thank you very much for listening.